Gavi Siddesh.
Good morning, sir. Good morning, sir.
Morning, morning. Can we start? There are
Yes,
only
sir.
79 have joined. Fundamental subject.
Sir,
Okay.
join after start ಮಾಡೋಣ sir.
Okay. Okay. We last class we were discussing on some terminologies. Okay, and we discussed what a gene is, what an allele is, and we discussed what backcross is, what test cross. Did we discuss what test cross is? Okay, we did discuss a few things, and now after knowing some terminologies, we'll straight away get into the Mendel's experiments. Okay. Get John Mendel's. Are you able to see? Gavi Siddesh, ಕಾಣಿಸ್ತಾ ಇದೆಯಾ?
Yes, sir. ಕಾಣಿಸ್ತಿದೆ sir. ಕಾಣಿಸ್ತಿದೆ.
Okay. Let's discuss about Mendel's experiments. Okay. So even before Mendel could start his experiments, he collected several varieties of garden peas. Okay, he collected different varieties of garden peas by some Sataivam. 2n is equal to 14. Okay, that means there are 14 chromosomes. Again, please remember, we generally don't call it as 14 chromosomes, we call it as 7 pairs. We call it as 7 pairs. Okay. Why 7 pairs? I told you, one set of chromosomes comes from the mother, the other set of chromosomes come from the father. Is that clear? Two gametes fuse to form a zygote. Each of the gamete has half the chromosome number. Each of the gamete has half the chromosome number. And the two gametes fuse to form. So what we have to write is the male gamete has n number of chromosomes. The female gamete in the form of egg has n number of chromosomes. They would fuse to form the 2n state. So I said 2n is equal to 14. So n is equal to 7. This is equal to 7. So the diploid chromosome complement is restored after the two gametes, male and female gametes, are fused. Is it clear? Well, when the chromosomes are coming together, one set of chromosomes coming from the father, one set of chromosomes coming from the mother, they fuse together to get the diploid state. Okay. Along with the chromosomes, even genes come. Along with the chromosomes, last class we did discuss, you have chromosomes like this, and on the chromosomes we have the genes arranged. Okay. So we have one set of genes on a chromosome. Say, for example, I said n is equal to 7. Yeah, 1, 2, 3, 4, 5. So if you take chromosome number 1 from the female, chromosome number 1 from the male, let us say if they have 100 genes, the male chromosome has 100 genes, the female chromosome also has 100 genes. Okay? Now, for example, this gene. Now, if this gene is for flower color, on the chromosome contributed by the male, this gene will also control flower color that has come from the female. Is that clear? And this is called as a locus. It's called as a locus. What is this locus? Position of a gene on a chromosome is called as a locus. Position of a gene on the chromosome is called as a locus. Is that clear? Now, these two are referred to as homologs. They're called as homologous. What do you remember homologous chromosomes? I think you have studied in cytogenetics. Can you recall? Can you recall what homologous chromosomes are?
Same set of chromosomes.
Same set of chromosomes, Allah.
So, identical chromosome in diploid cells.
No, actually, chromosomes that are identical in their size, shape, position of the primary construction, number, and order of the genes. Did you get it? Homologous chromosomes are those that are identical in their size, shape, structure. When I say size, length of the chromosome, number of genes, and the order of the genes as well. So if there are 100 genes on this chromosome, on its homolog also it will have 100. And if the order is A, B, C, D, E, F, G, H, here also the order would be A, B, C, H. Is it clear? That means to say that in an individual, at any given point of time, if the individual is sexually reproducing species, if the individual belongs to a sexually reproducing species, the individual will carry 2 copies of a gene. The 2 copies of a gene could either be identical. What is 2 copies of a gene? They are nothing but alleles. Alleles are nothing but forms of a gene, isn't it? If the 2 alleles are identical, then it is called as homozygous. If they are different, that is called as heterozygous. Did you get it? We have something called as homologous. What is homologous? Homologous, we refer to chromosomes. Homologous, we refer to chromosomes. Chromosomes that are identical in terms of their length, position of the primary construction. The primary construction is nothing but the centromere. Let us say the centromere. Secondary construction is nothing but the satellite body. So here also you'll have the satellite body. Number of genes, let us say this is 100, this is also 100. If there is a gene for flower color here, there's a gene for flower color here as well. Is it clear? This is homologous. Okay. Now coming to homozygous. Homozygous. See here. This is zygous. Zygote. A zygote resulting from fusion of identical alleles. Homo is identical. Now, let me say. Now, if the female plant and the male plant, let us say, designated male plant, both of them are producing yellow flowers. So, they carry an allele for yellow color. So, what happens? Both of them are yellow. So this is homozygous. Instead of yellow, if this is green or say white, then this is heterozygous. A zygote resulting from the fusion of non-identical alleles is heterozygous. A zygote resulting from the fusion of identical alleles is called as homozygous. Any doubts in this? Is it clear?
Yes, sir.
Homozygous is a condition wherein an individual at a particular locus, both the alleles are identical. How does it result? It results from a zygote first. I told you, all sexually reproducing organisms begin from a single cell zygote. If the zygote has resulted due to the fusion of identical alleles, then that individual is homozygous. If the zygote results from fusion of non-identical alleles for a character, then it is heterozygous. So what did Mendel do? Mendel had collected several varieties of P. Okay, he had collected several varieties of P. He carefully selfed them. He selfed the different varieties of P. Why? Why did he self? Now, primarily, garden peas is a self-pollinated species. To confirm whether they are homozygous. Now, for all genetic studies, please remember, please make a note as well. For all genetic studies, it is preferable to use pure breeding strains. Pure breeding strains. When I say pure breeding strains, homozygous individuals are considered pure breeding. Homozygous individuals are considered pure breeding. Did you get it? Did you get it?
Sure.
Homozygous individuals will breed true. For example, let us say this is a homozygous individual. Let us say this individual is tall. Now, if you self this, all the progeny, something like this, you have a plant, the genotype of the plant for plant height, let us say it's tall one, it's a tall one. Now, it is homozygous. So, if you self this, what is selfing? Put a bag on this plant. Don't allow foreign pollen to contaminate. So, you will harvest a number of seeds. When you rise those seeds, all of them will develop into tall plants. When will that happen? If only this is homozygous. If this were to be heterozygous, they will not breed true. Heterozygous individuals will not breed true. Is it clear? Is it clear? Heterozygous individuals will not breed true. Homozygous individuals will breed true. So what did Mendel do? The first thing that he did was he collected a number of varieties of P. He sent them continuously for 2 years. Made sure that all his parental strains were pure breeding. Pure breeding andre ಏನilla. Pure breeding andre ಏನು? Homozygous. Clear? Is it clear?
Yes, sir.
Okay. The first thing that he did was he collected a number of parental lines, sent them continuously for 2 years, made sure they were all pure breeding, and he used them as parental lines. The first character that he studied was plant height. But please remember, to study the inheritance of any character, you will need 2 parental strains. You will need 2 parental strains that are contrasting for a trait. So what did he do? He wanted to study the inheritance of plant height. So what did he choose? He chose a pure breeding tall plant as female, crossed it to a pure breeding dwarf plant. Does me? Is that clear? Then what did he observe? When he made a cross between these two, the resulting F1 plant was tall. Then this tall plant, no, you don't get only one plant. Please remember. How many, say for example, what do we do? You have a tall plant, it produces a number of flowers. One of the flower you emasculate, remove the male reproductive units, so that flower becomes female, get pollen from the dwarf plant, which is considered as male. You have the F1 plant. I'm sure all of you must have seen pea pods. Nodidira? Bari one seed irutta adralli?
No, sir.
Sumar irtavalva?
Yes, sir.
Yeah. So you'll have a number of seeds. Okay. Now they're intercrossed. They are intercrossed. See, the F1 is tall. You don't have only one seed. You have a number of seeds. All of them are tall. All of them are tall. Now that is why I told you the F1 is always tall. The F1 is tall. Irrespective of whether you use tall as female or dwarf as female. What is tall as female, dwarf as female? It is a set of reciprocal clauses. Irrespective of whether a tall plant was used as female or dwarf plant was used as female, the resulting F1 was always tall. So what is the conclusion that Mendel gave? The F1 generation has uniform phenotypic expression. And that is called as the law of uniformity. Law of uniformity. All the resulting F1 individuals have the same phenotype or uniform phenotype. Did you get it? Did you get it? Irrespective of whether you use tall as female or dwarf as female, when I said tall as female, obviously dwarf is male. If you use dwarf as female, then tall is male. Is that clear? So the resulting F1 progeny were found to be always tall. So this is called as law of uniformity, irrespective of whether you use tall as female or tall as male. Progeny derived from a set of reciprocal clauses between tall and dwarf produce F1 progeny that were identical in their phenotype. All the individuals were identical in their phenotype and all the individuals' phenotype was tall. Is it clear? Such tall individuals were crossed among themselves. Such tall individuals were crossed among themselves. What did they get? They got tall plants and dwarf plants. Tall and dwarf plants. How did they segregate? How did they segregate? Three by four. 3 out of every 4 plants were tall. 1 out of every 4 plants were dwarf. Is that clear? 3 out of every 4 plants were tall. 1 out of every 4 plants were dwarf. Now, let us give some explanation to this. You see here, I said, for every character in an individual, in a sexually reproducing individual, for each character, you have 2 copies of the gene. Let us say, for simplicity, this is tall. This is dwarf. Okay? Now, this is parent 1. This is parent 2. Now, now this will produce an egg. Egg is a gamete. So, what is the gamete? The convention is gametes are always encircled. Gamete should be encircled. Arthaita? Gametes are encircled. So this is another gamete. Now these 2 gametes will fuse to give rise to a heterozygous individual. Is it clear? Is it clear?
Yes, sir.
Okay. Now let us see how things go about. Now this is, I told you, the F1 tall plants were crossed amongst themselves. How did they cross? So, is it clear? Now, how many different kinds of gametes will this individual produce?
2, sir.
So, this is a female. This is a female. So, what is a female? These are the two kinds of eggs that this individual produces. These are the two kinds of eggs. Now, what about the two kinds of pollen produced by this? Did you get it? It's something like this. Please, you'll have to imagine a plant. Okay, you have a plant. Okay, you have mastered the plant. Let us say this is a flower. Onto the flower, you get the pollen. The pollen are in the form of capital T and small t. The eggs are in the form of capital T and small t. Is it clear? Is it clear? Yes or no? Okay, I'll put it this way. There are two eggs. Eggs that carry an allele for tallness or an allele for dwarfness. Pollen grains that carry allele for tallness or allele for dwarfness. Now, when they are sitting on the stigma, this can either fuse with this or with this. Similarly, this can fuse with this or this. So that is why random union of these gametes. Random union of these gametes will result in these four individuals. Is it clear? What is the phenotype of this individual?
Tall.
Tall. What is this individual?
Same, sir.
Tall. What about this one? This one? Now, don't restrict your understanding to only 4 plants. There could be large number of eggs in a plant. Did you get it? Large number of eggs in a plant. Large number of pollen in a plant. So, large number of eggs can unite with large number of pollen. And for every 4 plants, 3 will be tall and 1 would be dwarf. Is that clear? For every 4 plants, 3 will be tall. Now, say for example, if I have 400 plants from this, how many plants are expected to be tall?
300.
300 are expected to be tall and 100 are expected to be dwarf.
Dwarf.
Clear? Mendel did not stop here. Mendel did not stop here. What did he do? He wanted to confirm his results. He wanted to confirm his results. How does he confirm? Confirmation is in 2 kinds. Confirmation is in 2 ways. So here you have pure breeding tall, cross to pure breeding dwarf, the resulting of fun. Okay. To confirm the genetics of it, right? So what did he do? He performed a test cross. What is a test cross? Test cross is crossing the F1 with a recessive parent. Now, the term is coming as recessive. So, what is the other word for it? The opposite word for it? Then you have something called as dominance. Or recessive. Now, how do you define recessive and dominance? When you made a cross between 2 contrasting forms, the F1, some form is expressed. So you made a cross between tall and dwarf plants. Tallness is expressed. That means to say that tall is getting expressed by suppressing the phenotype dwarf. Is that clear? Is it clear? The trait that expresses itself in the F1 is called as dominance. Is that clear? And the allele governing such form is called as the dominant allele. the form that is suppressed in the F1. The form that is suppressed in the F1, but it reappears in the F2. It reappears in the F2. With what frequency? It reappears in the F2 with a frequency of what? So when you make a cross between two contrasting forms, only one of the form is expressed in the F1, the other form disappears. What are the forms that we're talking about? Plant height, tall, and dwarf. When you make a cross between tall and dwarf, the F1 is tall. That means say that the other form, alternate form, has disappeared. But it reappears in 1/4 of the F2. So the form that disappears in the F1 but reappears in the F2, how much? 1/4 of the F2 is referred to the recessive allele. Did you get it? Is it clear?
Yes, sir.
Yes, sir.
When you are making a cross between two contrasting forms, the phenotype that is expressed in the F1 is called as the dominant form. The form that disappears in the F1 is called as recessive. But you don't stop there. The form that disappears in the F1 but reappears in 1/4 of the F2 is called as recessive. Is it clear? Is it clear? The form that disappears in the F1 but reappears in 1/4 of the F2 is called as the recessive form. So that is what is dominance. But we'll get to know what dominance is a little later. Okay. So this is 1/4. Now, Mendel wanted to confirm this. So what did he do? It has crossed. So what is recessive here? What is recessive? The trait that disappeared in the F1 is recessive. So what did Mendel do? He crossed the F1 with that of the dwarf plant. What do you get in the F1? See here? The F1 produces two kinds of gametes. The dwarf plant produces only one kind of gamete. If they unite, this is tall, this is dwarf. So this is the test cross progeny. Okay. This is a monohybrid. There's a monohybrid. What exactly do we mean by monohybrid? An individual that is heterozygous for one locus is a monohybrid. Mono, single. Hybrid, heterozygous. An individual that is heterozygous for one locus is a monohybrid. This is a monohybrid test cross. This is a monohybrid test cross. What is this then? What is this? This is a monohybrid cross. You are crossing two monohybrids. When you cross two monohybrids, what do you get? You get the F2. Is it clear? Now you'll have to get acquainted with all these terms. I'm sure you must have studied in PUC, but the way we deal with the subject here at the graduate level is completely different. Is it clear? I may be repeating the same thing. It could be boring a bit. Is it clear?
Yes, sir.
Okay. Now, one is a test cross. What is a test cross? A test cross segregates in the ratio of 1:1. Clear? Now, Mendel, what he did, he hypothesized. This is an F2, right? When you make a cross between two heterozygotes, what you derive is an F2. What is the F2? So what Mendel did was, he wanted to confirm the inheritance of these plants. So what he did, he selfed all of them. Now this is the symbol for selfing. A circle with a cross mark. Circle with a cross mark is selfing. So what did he do? Mendel selfed each and every individual of the F2. Each and every individual of the F2. Okay? Then, what he observed was, we know this is tall, this is tall, this is tall. So 3 out of every 4 plants are tall, 1 out of every 4 plants are dwarf. What Mendel hypothesized, expected in the F3. Now this is your F2. When you self all of them, you will get the F3. So what Mendel did was, he selfed all the individuals in the F2 and he tracked how the trait is behaving in the F3. What he found was one third of the F2 tall plants, one third of the F2 tall plants were observed to breed true. Were observed to breed true. 2/3 of the F2 tall plants, 2/3 of the F2 tall plants segregated and behaved as if they were F1. This one. They behaved similar to that of F1. And 1/4 of the dwarf plants, all of the dwarf plants produced progeny that were dwarfs. When I say be true, you sell this individual, all the seeds that you get, you raise them, all of them are found to be tall. When you harvest seeds from this plant, they will segregate as tall and dwarf. This will also segregate as tall and dwarf. This plant will breed true for tallness. So what is the conclusion of Mendel? He expected, when he selfed the F2, he expected the results in the F3 even before he conducted the experiment. He expected all the dwarf plants should breed true, 1/3 of the tall plants will breed true, and 2/3 of the tall plants will segregate. And that is what he observed. Mendel was the only scientist who studied or went up to F3. The other scientists stopped at F2. Is it clear? And that is the reason why Mendel was successful in his experiments. Now, based on the results that he saw in this particular thing, he came out with a law or a rule. That is called a law of segregation. Law of segregation. What is that law of segregation? When you make a cross between two individuals contrasting for a particular form, so this is tall, this is dwarf, F1s, they're all tall. In the F1, the two contrasting alleles are staying together. An allele for tallness, an allele for dwarfness are staying together. For how many J's? For one full generation. The contrasting forms of a gene, though THOUGH, though they stay together for one entire generation, they do not blend with each other. They do not mix with each other. Instead, what happens? During gametogenesis, they disjoin and move into different gametes. Now, let us see how exactly it happens. Let us say this is a pure breeding tall plant. I was talking about chromosomes and a locus here, cross to a dwarf plant. What happens to the F1? You have capital T, small t here. Clear? This is the F1 hybrid. Now, they will separate, the chromosomes will separate. Please go back to meiosis. Homologous chromosomes disjoin. Where do they go? Disjoin and move into different gametes. One gamete carrying the allele for tallness, the other gamete carrying an allele for dwarfness. And if they unite at random, what do you get? The same thing. This is also the same thing. You have a chromosome here, you have a chromosome here. You mix them together, this is what you get. Is it clear? Is it clear? Yes?
Yes, sir.
Okay. Though the contrasting forms of a gene here, though the contrasting forms of a gene stay together for one entire generation, they do not blend with each other. Instead, they separate and move to different gametes. They separate and move to gametes. Where these gametes carrying contrasting forms of the trait unite at random, you'll get 3 tall and 1 dwarf. This is law of segregation. It's also called the law of purity of gametes. Law of purity of gametes. You see here, this is an egg. Egg is a female gamete. This is an egg. This is a female gamete. This is a pollen. Pollen is a male gamete. The gametes carry either an allele for tallness or dwarfness. Either an allele for tallness or dwarfness. A gamete will not carry both the alleles. So therefore, it's called as law of purity of gametes. A gamete is always pure for a particular trait. Artha aita? Artha aita? So what is law of segregation? So when you make a cross between 2 contrasting forms, for example, a pure breeding tall plant into a dwarf plant, the resulting F1 is tall. In the F1, the contrasting forms of a trait stay together for one entire generation. Though they stay together for one entire generation, they do not blend with each other. Instead, they separate, disjoin, and get into different gametes. And when these gametes unite at random, you'll get a 3:1. Is it clear? Is it clear? Have no.
Yes,
Hello, Malput, try now.
Yes, sir.
ಕೇಳ್ತೀವಿ ಸರ್ ಕ್ಲಾಸ್.
Is it clear? Is law of segregation clear?
Yes, sir.
Any doubts? Now here we have discussed about one gene. On this chromosome there could be several genes. Each of the gene will behave like gene for plant height itself. There could be an allele for something else. There could be hundreds of genes on the chromosome. Each of the pair of genes will behave as if they are like contrasting forms for plant height. Okay, sir, is there any reason why the dominant and recessive genes do not blend with each other? So genes per se do not blend. Genes do not blend. That's as simple as that. They do not blend. Okay? If they were blending, then you wouldn't have got tall plants in the F1. Is that clear? Tall is dominant over dwarf. Okay? If tall is equal to dwarf, then you would have an intermediate plant height. Is it clear? There was a chat. Anything else? Okay. I will stop here if you have no doubts. If you have doubts, we can spend some time. Is there another? Gavi Siddesh?
ಸರ್
ಏನಾದ್ರು ಇದೆಯಾ ಡೌಟ್ಸ್ ಇದೆಯಾ
ಇಲ್ಲ ಸರ್
ಕ್ಲೋಸ್ ಮಾಡೋಣ ಕ್ಲಾಸು
ಸರ್ ಅಟೆಂಡೆನ್ಸ್ ತಗೊಂತೀನಿ ಒಂದ್ ಸ್ವಲ್ಪ ನೆಲಕ್ಕೆ ಇಡಿ
ಹಾ
ಅದು ಸ್ಕ್ರೀನ್ ಶಾಟ್ ತಗೊಂತೀನಿ ಸರ್
ಹಾ ತಗೊಳ್ಳಿ one more question I guess Now you are most welcome to ask. You can stop me at any point of time. Okay, you can ask me any number of times. If you have no doubts, we'll meet next Monday between 12 to.
Sir, ತಗೊಂಡೆ sir screenshot.
Tondra?
ಹಾ sir.
Okay, Napa. Huh? Okay. Have a nice
Thank
time. Enjoy your festival. Ella ಊರಲ್ಲಿ ಇದ್ದೀರಾ?
ಹ್ಞೂ sir.
Okay, Napa. Huh? Okay.
Thank you sir.
Okay. Right.